Hello, this is a Chapter 15 lecture on immunology. And I'd actually like to begin with talking about the lymphatic system, which is covered in the textbook in the end of Chapter 13. And I've saved this because it integrates nicely with the immune system. So the lymphatic system plays two important roles. One, it has a role in recirculation, and for this, it transports excess interstitial fluid, which we call lymph, from tissues back to veins. Secondly, it has an important function in the immune system because it produces and houses lymphocytes for immune responses. So here is a diagram showing the relationship between the circulatory and lymphatic systems. So we previously talked about how arteries will have will fade into capillaries, and then the arterial end of a capillary will have fluid leaving blood vessels into tissues, and then from tissues there's going to be some uptake of fluid into the venous end of the capillaries. And what I'm showing you now is that there will also be fluid movement from the interstitial tissue into the lymphatic capillaries. And then from there, it can go into the bigger lymph ducts. The fluid will actually enter lymph nodes. And then from the lymphatic ducts, the lymph will be brought back to the venous blood vessels. So the lymphatic system and the circulatory system are connected. And again, I want to repeat that excess interstitial fluid will be picked up by the lymphatic capillary, sent back through the lymphatic ducts. It will pass through some lymph nodes and eventually be sent back to the veins for re-entering the circulatory system. So lymphatic capillaries are the smallest of the lymphatic vessels, and they're found in most organs. They have a very porous surrounding. So since lymphatic capillaries are very porous in terms of letting exchange of fluid, they pick up interstitial fluid. They can even pick up proteins, microorganisms, and fats. And these lymphatic capillaries are going to merge into lymphatic ducts. And the lymphatic ducts have very similar structures to the veins, where they have smooth muscle around them. So peristalsis can help the lymph move through the lymphatic vessels. And it also has the mechanisms for preventing backflow. And lymph nodes play a key role in filtering lymph. And again, I just want to show another diagram showing a tissue. Here we have in yellow showing the lymphatic capillaries, right? And they'll be picking up fluid from the interstitial tissue. And these lymphatic capillaries are going to feed into these bigger lymph ducts. And then eventually the lymph will be sent to lymph nodes, and then after it's been filtered by the lymph node, it'll be sent back to the veins. The lymphatic ducts are going to deliver lymph into the right and left subclavian veins, and this will help get the purified lymph back into the blood. So there are some important organs in the lymphatic system. These include the tonsils, the thymus, and the spleen. And these three organs, along with lymph nodes, are sites for lymphocyte production. And here, if we take a look, we see the spleen over here, we see the thymus, we see the tonsil, and then I mentioned that the lymph nodes are have a purifying aspect or filtering aspect in the lymphatic system, and these lymph nodes throughout the body. And when you're sick, often the doctor will be feeling the lymph nodes in your neck to see if they are swollen, and they often will get swollen during an infection. So if we take a closer look at the lymph nodes, we can see that there's these germinal centers, and the germinal centers are centers where there could be maturation of lymphocytes. And the lymphatic ducts will end up bringing lymph into the lymph node, where there'll be a filtering of, for example, microorganisms. If there's a virus or bacteria in the lymph, the immune cells of the lymph node will work to help purify and purify the lymph to remove these microorganisms. So now I'd like to get into the immune system. And there are two different branches of the immune system. One is called innate immunity. Innate immunity is the part of our immune system that prevents us from getting sick. It is nonspecific in that the innate immune system has the ability to recognize structures on the surface of common structures on the surface of microorganisms, and then to recognize those common surface structures and to make an immune response against them. So someone with a really good innate immune system will not get as sick as someone with a weaker immune system. Or sick, you won't get sick as often, right? So if someone who has a good innate immunity will get sick less frequently than someone who has a weaker innate immune system. The adaptive immune system is focused on, once you are sick, on getting better. So the adaptive immune system is where your body will mount a very specific response to a microorganism causing an infection. It'll react to specific antigens. These could be parts of a foreign protein or sugar or chemical that is on the surface of the microorganism. And surface is a very important word here, and we'll talk more about that in a little bit. And if you generate an antibody response, there will be a memory of it. So the next time that you are exposed to that same microorganism, your body will mount a rapid immune response, so you won't get sick. And that basically is what we call immunity. There's many different cell types that play a role in the immune system. And I would like to point out that the B cells and T cells play a role in specific immunity, and all the other cell types play a role in non-specific. But the specific are really the B cells and the T cells that play a role in specific immunity. The other cells are all going to be playing a non-specific role. So if we look at the innate immune system in closer detail, we have some physical barriers to infection. This includes skin. The skin has keratin proteins that make the skin a tight layer and prevent fluids from penetrating and going into the skin. And skin also has oil that helps to have organisms not penetrate us. And the skin also has, in the deeper layers, phagocytic cells that can basically destroy microorganisms. Secondly, when we wash our hands, we can remove dead skin cells by washing and rubbing. Skin cells that are old just fall off, and if there's a microorganism attached to a dead skin cell, that'll be removed as well. We have mucus, for example, in our throat and in our oropharynx, the nose, and this can trap pathogens. And then basically, if you, let's say, for example, you have some mucus in your mouth, that can bind to a microorganism and take it down to your stomach, where it'll be killed by the low pH of the stomach. We also have mucus layers lining the intestine, but basically, if a microorganism attached to the mucus layer, it can slough off and be brought to the stomach, and the microorganism can be killed. In the throat, we have, and from the throat going to the lungs, we have cilia that are beating up. They're basically like at the wave at the arena, where the cilia are basically beating and pushing up away from the lungs. So if there's a microorganism traveling in air to your lungs, if they hit the cilia will try to take them to the saliva and mucus in your throat So then they'll be brought down to the stomach, down the esophagus to your stomach, where again the pH of 1 in the stomach will hopefully kill the microorganism. So a lot of our immune cells have these toll-like receptors, and these cells are phagocytic. So in the gut we have M cells that have the ability to recognize common structures on the surface of microorganisms. And on the skin, we have Langerhans cells that basically have that ability to recognize common surface structures on microorganisms and to phagocytose them and then put them into a phagolysosome where they will be destroyed by degradative enzymes and degradative molecules. For chemical barriers of host defenses, we have the acidic pH of the stomach, which I mentioned can be pH 1 in the stomach. Skin is slightly acidic, and so is the vagina, and that basically inhibits the growth of many microorganisms. In tears and in skin, we have lysozyme, and lysozyme is a large enzyme produced by our immune system, and it's the extracellular enzyme that has the ability to destroy peptidoglycan, which forms the cell wall of bacteria. We'll talk about that more in a minute. And the immune system can also produce antimicrobial peptides, which are small peptides that have the ability to destroy microorganisms. And here's a picture of a peptide basically causing the blebbing of a membrane. It's destroying the membrane of this bacteria. In order to understand how bacteria react to the immune system, we need to review bacterial cell envelopes. So bacteria can be classified into either gram-negative or gram-positive bacteria. So gram-negative bacteria have two membranes, an inner membrane and an outer membrane. And the inner membrane is made up of phospholipid bilayer. The outer membrane has phospholipids on the inner leaflet and lipopolysaccharide on the outer leaflet. In between is a peptidoglycan layer. This is a sugar layer that is typically what we call the cell wall of bacteria. So the inner membrane of gram-negative is called symmetric, and the outer membrane is called asymmetric because on one leaflet we have phospholipids, and the other leaflet we have lipopolysaccharide. So lipo means lipid, polysaccharide means many saccharides, many sugars going out into the environment. So it turns out that the lipopolysaccharide is an endotoxin, and this endotoxin, LPS, if found in high numbers in the blood of humans or mammals, can basically trigger a septic shock response, which is when all the blood vessels in the body will vasodilate, leading to a loss of blood pressure. This can then lead to organ failure and death, and we'll be talking more about that. But I did want to point that out, that it is very dangerous having a gram-negative bacteria in your blood, or most gram-negative bacteria in the blood. And in contrast, gram-positive bacteria, if they're in your bloodstream, they are not as dangerous as the gram-negative because they are lacking the endotoxin, lipopolysaccharide. So gram-positive bacteria, in contrast to gram-negative, they have only one membrane, which is called the plasma membrane, and then they have a thick peptidoglycan layer, in contrast to the very thin peptidoglycan layer of gram-negative bacteria. So the immune system can see what is on the surface of a gram-negative and gram-positive bacteria and know that there's a gram-negative or gram-positive bacteria in the body. On a gram-negative bacteria, the immune system will see lipopolysaccharide. And there is a toll receptor that can detect this particular molecule. In contrast, there's another toll receptor that can see peptidoglycan. And then the immune system can make an appropriate response for a gram-positive bacteria.